Hello, scientists. So today we are going to learn about experimental variables. When it comes to experimental design, I actually believe that there's a lot of art in designing an experiment. And a good experiment doesn't have to be difficult. Some of the best experiments in history were actually pretty simple, but they were really well designed. And if you can design a good experiment, that basically means that your results are going to be stronger. So let's jump right in. Let's start with our learning targets. Okay, so first of all, I want you to be able to identify and describe the four main variables in any scientific experiment. This could be done in a couple different ways. I might give you an experiment and you have to tell me what the variables are, things like that. Next, I want you to identify whether controls were used, and if they were they positive or negative? It's okay if you don't know what that is right now, you will. Next, I want you to be able to describe why a constant variable is so important in an experiment, and we'll talk about that. I want you to be able to choose variables in your own experiments or examples to keep constant in order to strengthen those results at the end of the experiment. And then finally, we will be working on becoming really good critical readers of science. So if I give you a scientific experiment in literature, you might say, wow, like this is good, but what if they had done this? And really kind of identify some flaws in some experiments. So those are our goals for today. Little cartoon here for you. I love Calvin and Hobbes. I love what he says here. In my opinion, we don't devote nearly enough scientific research to finding a cure for jerks. So a little funny thing for you. Okay, first variable. So you should be taking some notes here. Is called the independent variable. Sometimes you will see me write it as IV. So that's shorthand for independent variable. So this is what you, as the scientist, are changing in the experiment. Okay, so as the scientist, you are going to be affecting something happening in the experiment. In any experiment, it's really, really, really important to have a single independent variable. So when we start designing our own experiments, this is hard. This is hard to become good at. And I might say to you, well, you have more than one independent variable. And then we don't know which one is causing the change. So we'll talk more about that. So here's an example of an experiment. I've got 4 plants. They're going to get different amounts of fertilizer. So this one gets no fertilizer, 2 drops of fertilizer, 4 drops of fertilizer, and 6 drops of fertilizer. So what is the independent variable on this one? Well, it is the amount of fertilizer, okay, or the number of drops of fertilizer. When you go to graph your data, let me go back to a pen here. Sorry. Classic. I don't know how to do that. When you go to do this on a graph, because you will be graphing your data, okay, I want you to know, so let's say that this is your graph here. Your independent variable will always go on the x-axis, which is the bottom axis, always. The only time that it doesn't is if you have time, and I'll get, like, time is one of your variables, and I'll explain why that is. Okay, so here's an example of this experiment on a graph. So you can see we said that the independent variable on this one was the amount of fertilizer, okay? And that is exactly what it is on the graph. So that is your independent variable. Let me clear this off so that we can do the next one. The next variable I want you to know is called the dependent variable. Okay, so this one, if you see it in shorthand, will be DV, dependent variable. So this is what you as a scientist are going to be measuring. So hopefully if your hypothesis is correct, you will see a change based on what you did with the independent variable. So if we go back to that other experiment, we change the amount of fertilizer, and hopefully the one with the most fertilizer will grow the most, right? So that's kind of what we're looking for. So what I'm going to measure in this example are lots of different things, okay? So I can measure the height of the plant. I can measure number of leaves, things like that. So what this does, though, is it's very important that you as a scientist find a way to measure what's happening from how you changed what was happening. So on the graph, right, like we did before, the x-axis is your independent variable, which we already talked about. So your dependent variable then would go on the y-axis, which is on this side of the graph. Now, when it comes to dependent variables, you can have a lot of them. You do not have to cut it down to a single one. As a matter of fact, I would suggest, taking data on a lot of things. So here is our graph again. Okay, and as you can see, we have amount of fertilizer on the x-axis, and we have plant height after 2 weeks on the y-axis. So perfect. That's how it should be. All right, next, let me erase this again, is something called a control. And we'll talk about this in class because this is probably one of the ones that stumps students the most. A control is something in which you have a positive or a negative known outcome. So this is kind of what we call quality control, right? So you're going to use this to compare the groups that you made changes to. So for example, a negative control, if I'm studying a new drug, I might give half of the group a saline shot. So they're still getting a shot. They still think they're getting the drug, but it doesn't actually have it in it. So they're not getting what I'm doing. A positive control, well, those are experimental groups. So those are the groups that are getting it. It's also sometimes on a test, like if I'm taking a test for COVID, they will also run a known positive sample next to mine. Because if that one doesn't come up positive, then they can't trust their results. They're always used for comparison. Here's the example of the test I was telling you. So, right, so if you look here, hope you don't mind the pens, this is what a positive result should look like. So let's say we're talking on this one about influenza, okay? So we're doing a blood test and we put the serum in here and it turned red, and that indicates that the patient has influenza. Here's a negative result, it should not turn red. And here's my patient sample. So you automatically know, since these are equal, my patient is negative for flu. However, what if my positive sample did not turn red? If that were the case, and this was also not turning red, I still could not trust those results because my test didn't work. So we'll talk more about that in class for sure. All right. Like I said before, this is just reminding you, if your controls are not what you expect, you cannot trust your results. Okay, so we'll go into that more in detail when we're designing our experiments. The last one I want you to know is called a constant or controlled with an ED at the end. So it's different from a control versus controlled. I like to call them constant variables because it helps kind of keep it separate, I guess you could say. Oops. These are variables that you're going to keep the same for every group, right? And so think about our plant example, okay? We changed the amount of fertilizer, if you remember. Okay, so we changed how much fertilizer each plant was getting. But there are some things that still could affect plant growth, and we want to keep those the same for every group so that they don't affect the outcome. I want you to pause, kind of think about some things, and then come on back. Okay, so I asked you to think about some constant variables. Some good ones would be amount of light. It could be type of light. It might be how much water they get besides the fertilizer. It could be the age of the plant, right? You want to use the same species of plant. You want it in the same container with the same amount of soil. So it is through the constant variables that the artistry of designing experiments really comes about. Because the more that you can control for, the stronger your experiment will be. Basically, what a constant variable does is it avoids anything else from affecting the outcome, so that if you do see a difference, you can say with pretty strong belief that it was because of the independent variable. Like I said, this is the art of science, okay? I'm going to give you an example, and you're going to answer this and be prepared to share it in class. I wanted to see if the amount of sleep students were getting affected their test scores. So I want you to take a moment and answer the following questions. What is the independent variable? What is the dependent variable? Is there a control? If there is it positive or negative? And then finally, the most important thing, I think, are there things that you can keep constant? So that's it for today. Hope you guys have a fantastic night, and I will see you guys in class.